Vinash, yeah. Hello. Now we are live, sir. So we can start now. Um, we are in the third session of the workshop, national workshop, and we are very fortunate to have the stalwart personalities in the field of mycology with us. I heartily welcome our honorable chairman for the session, Dr. Yashpal, sir. I also welcome Dr. Navneet Singh, sir, and my welcome is also due for our honorable president of MSI Mumbai unit, Dr. Deshmukh, sir, and all the dignitaries in the Zoom meeting as well as on the platform of YouTube. We have very good response for all the sessions. Just now we have completed the exercise, and that exercise was for one day assignment in the first session, and it was the second assignment in the form of assessment through the quiz. And it was very tough quiz, and the scores itself are reflecting that we have maintained the quality of the quiz and the questions placed in there. So it was the huge response from the students, those who are in the PG, UG, and research categories. At the same time, many of the teachers have also contributed in the activities. So we are fortunate to have such a galaxy of renowned persons for this workshop. So today we are here with us as the chairman of the session, Dr. Yashpal Sharma, sir. The speaker for this session, Dr. Avneet Pal Singh, sir, and our honorable chairman, president of MSI Mumbai unit, Dr. Deshmukh, sir. To begin with, I must introduce the chairman for the session, Dr. Yashpal Sharma, sir. Begin with the request to our honorable chairman, Dr. Yashpal Sharma, sir, to accept our invitation to chair the session. And I also invite Dr. Navneet Pal Singh, sir, to interact with our students and the researchers. In the initial stage of the session, let me introduce our chairman for the session, Dr. Yashpal Sharma, sir. We are very fortunate to have such a stalwart personality with us. While going through his biodata, it was very difficult for me to sum up his entire achievements in a very short period of time, which is given to me for introduction. Dr. Yashpal Sharma, sir, is working as professor and coordinator for UGC Special Assistant Program. He is working at the Department of Botany, University of Jammu, and I think his dynamism is reflected by his contribution in variety of the fields. As far as his subject area and research area is concerned, he is devoted. I think he is a devotee for the field of mycology and plant pathology. He is also contributing significantly in the mushroom diversity studies, ethnobotany, and especially ethnomycology. Through the 24 years of his service, he has produced more than 12 PhD students, 14 MPhil students, and one PDF fellow is also to his credit. Presently, 8 students are working under him, and 3 research projects are going on. While counting the fellowships and awards honored to Dr. Yashpal Sharma, sir, I think it's very difficult for me. I'm once again asking excuse to Honorable Chairman that I could not go through the entire biodata. But let me count the feathers in his crown. He is honored with the special recognition as the Leadership for Academician Program, LEAP, which is the scheme of MHRD, Government of India, to undertake leadership training programs at ISER Kolkata and Singapore in the foreign category. This was achievement in the recent period, that was January 2020. He is also honored with the SK Memorial Ashok Memorial Award. Then he is the person having Professor H.C. Dubey Outstanding Young Scientist Award in the year 2010 by Indian Society of Mycology and Plant Pathology, Udaipur. He is Professor P.N. Mehta Young Scientist Award winner in recognition of the outstanding contribution in the plant sciences. Then he is also honored by the Young Scientist Award in the year 2003 by Jammu and Kashmir State Council for Science and Technology. I think these awards are just the recognition of his contribution in the field of mycology. He is a senior research fellow for CSIR in the year 1996, way back. And as far as the organizations of Dr. Yashpal Singh is concerned, he has organized almost 10 different programs like conferences, seminars, and workshops. And those workshops and conferences were the activities of the very high cadre. He is also the member of editorial board for very reputed publications like Springer, elsewhere, Trivedi publication, and other reputed journals. Regarding the publication, I must congratulate our honorable chairman for this session for having the huge contribution reported in different research articles. He has published 2 books, 38 research papers in the Scopus indexed journals. He has also published 63 papers in other journals of the national and international repute. He has also contributed 16 chapters in different books. As far as his contribution in the field of guidance to the students and researchers across the India and world, there is long list of lead lectures, invited talks, the speeches in national and international conferences, seminars, and workshops. I must congratulate the entire fraternity of the researchers who has joined this workshop and the students, because today we have very good opportunity to hear the words of wisdom from the very renowned personality from the mycology, Dr. Yashpal Sharma sir. While moving ahead, today's guest speaker who will enlighten the entire students and the researchers on a very specific field of mycology, Dr. Avneet Pal Singh. He is the very dynamic and enthusiastic personality from the field of mycology and contributing a lot in the Mycological Society of India. He is working as assistant professor in Department of Botany, Punjabi University, Patiala, and he is basically from the botany and completed his doctoral research in the field of mycology. He has described 160 taxa. This is very huge contribution, sir. I must thank to you, including 2 new genera and 18 new species of wood rotting fungi from the Himalayan range. He has more than 30 years of teaching experience. He is having the 2 awards to his credit. I must say that the best paper in technical session in the 2nd Asian Congress of Mycology and Plant Pathology was the beautiful award and recognition for his research. Sorry. He is also awarded with the University Medal for standing first in the post-graduation in botany. He is a member of different professional organizations. His research contribution is highlighted, and 3 students have benefited with the PhD degree under him. He has submitted 1 thesis recently, and 4 students are registered for the PhD under his able guidance. 2 MPhil students have received their degree, and many MPhil students, MPhil and MSc students are working under his able guidance. He has completed many research projects, and that project significantly he has completed the research project under the banner of DST. He has published 52 research papers in different national and international journals. Other contributions are a huge list. So instead of wasting time, we will come to know the complete personality of the Avneet Pal Singh sir with his talk on the specialized topic of mycology. May I now invite Dr. Avneet Pal Singh to start his deliberation and enlighten our complete fraternity of the teachers, researchers, and the students. Please, Avneet Pal Singh sir. You can share your PowerPoint, sir.
Thank you very much, sir. At the outset, I am extremely thankful to the entire team of Shri Shivaji Science College, Amravati, It's always been
Michael.
a sort of extended family to be with the friends from Amravati. And I still remember that 2012 MSI meeting, which I attended for the first time after joining Botany Department, Punjabi University, Patiala in 2011. And from that, it's been always an honor, a sort of homely feeling to interact with the friends from Amravati. And it's indeed a matter of honor to present one's work, or to speak on a topic in presence of the honorable chairman, Professor Yashpal Sharma ji, who has been just like an elder brother, an elder to me. And the kind of affection and the kind of support which he has been extending throughout is marvelous. And whenever we visit Jammu, the Jammu is just like that. I am visiting my family and a part of that only. So it's really indeed an honor to be with all such personalities, and in presence of such stalwarts. I'll just try to summarize whatever I have learned over the last few years under the able guidance of my teachers. And so, with the permission of Honorable Chair, I would like to start with my presentation. Uh, Thank you.
Yes, Abhijit, please. All
Thank
the
you, sir.
best. All the
Thank you, sir.
best.
Thank you, sir. So, the topic which I have selected for today's workshop, for today's lecture, that pertains to a field on which we have been working for the last few years. Initially, I started my studies on this important group, which we refer to as wood rotting fungi, the rhizopinnate ones, and gradually we shifted to this poroid fungi, which are even more fascinating group of these fungi. So the topic is systematics and diversity of polypores, with particular reference to Northwest India. So this lecture is divided into two parts. In the very first part, I would like to focus on the various characters and techniques which we use to work upon for their morphological studies. And the second part, I'll share with you some of my personal experiences in the form of certain descriptions which we have which we have described on the basis of these studies. Now, let us begin with a brief introduction what we have for today's group. Now, polypores, they are also called as poroid fungi. They are also known as polypores. They are also called as the wood decayers. Now, this group of fungi, as you must have listened to the morning session also, it is another important group of fungi within phylum Basidiomycota. The subphylum is Agaricomycotina. If we go by the latest concept, the class is Agaricomycetes, and these constitute one of the major group of wood rotting fungi in the forest ecosystems, and they contribute significantly towards the recycling of different types of organic matter. Now, if you look at these fungi, as all of us are aware that in Basidiomycota, we have the formation of beautiful fruit bodies or fructifications. Those fructifications, they are referred to as basidiocarps. And these basidiocarps are always, they are also formed in these polypore or poroid fungi. We have to take into account different characters, and these fungi, they are unique in having annual to perennial fructifications. Now, these fructifications, they may be closely appressed to the fruit body, to the substratum, and these closely appressed fructifications, they are called as rhizopinnate. They grow unilaterally. They grow along the substratum. They are having only that sort of appearance in which they are closely appressed to the substratum. From this rhizopinnate nature, Some of them, they gradually become reflexed. Their margins, they become somewhat separated from the substratum. And finally, ultimately, we have the formation of pileate basidiocarps, where typical pilei they are formed. And this particular group of fungi, they are having a unique gymnocarpic type of developmental pattern. Being students of botany, we study mycology, and there we always study that if you look at the evolution of the fructifications in fungi, they have evolved from gymnocarpic to angiocarpic mode through hemiangiocarpic pattern. This is with reference to the kind of hymenium which is under protection. Gymno means exposed. So those fructifications where hymenium, the spore producing portion of the fructification, is not covered with any protective covering, that is called as gymnocarpic development. So in these fungi, the hymenium is always exposed. It is not protected with any of the protective structure. And in polypores, the hymenium or the spore producing zone that lines the tubes, there is a formation of unique tubes in the fructification, and the lining of that tubes, that is made up of this hymenium. And these tubes, they open to the outside through fine openings, which we call as pores. And this is on the basis of these pores that the group has been named as polypores, because they have unique openings named as pores through which the spore dispersal that is taking place. So these are the fungi which are very unique in having this set of unique features. Now, since we have studied that there is a range from resupinate to pileate forms, now the pilei, they are highly variable in the shape. Shape is extremely variable. We will study some of the prominent shapes later on. Then these pilei, they may be formed either solitary or in groups. So that particular situation that prevails in these fungi. Now, if you look at these fungi where we have the formation of pilei, there is an upper surface. The upper surface is called as abhymenial surface. And the lower surface where we have the pores, that is called as hymenial surface or the hymenophore. These two surfaces, they are also highly variable. All these features, they are of importance when we undergo the taxonomic studies, when we take up the taxonomic studies of these wood rotting fungi. So the abhymenial surface, it ranges from smooth to tomentose through the variation of different features. It can be zonate to azonate, or it may be sulcate, where some sort of cracks regularly or irregularly they are appearing. On the other hand, the lower surface, which we refer to as hymenial surface or hymenophore, there we have pores, and these pores are highly variable in their shape. So such fructification bearing fungi, members of Basidiomycota, they are referred to as polypores or poroid fungi. If you look by the classification of that has been adopted in Kirk et al., that is the dictionary of fungi, these fungi, they have been placed under 17 families, and those 17 families are grouped into 7 orders of class Agaricomycetes within phylum Basidiomycota. Now there are two other aspects which are associated with these fungi. These fungi, they are responsible for causing different types of rots. Chiefly, the two important ones, they are white rot and brown rot, and they are responsible for the rot of both coniferous as well as broad-leaved tree species. So thereby, they play significant role in the forest ecosystem. Secondly, some of these basidiocarps, some of the members of polyporoid fungi, they have been used extensively in the traditional medicinal system. Ganoderma is one such example which has been exploited like anything. Trematis is another such species. So these fungi, they have also been now exploited for their medicinal properties. So this is the sort of group which we will be discussing in today's lecture during this session of the workshop. Now, before proceeding with what we have to discuss, let us briefly have an idea how this group it came into existence. Linnaeus, whom we all study whenever we have these studies on systematics, now, he was the first person who has used the term or the epithet Boletus. So, this was the first word which was used for all the fungi which are having tubes or pores on their hymeneal surface. Now, he has taken this concept of genus Boletus for those members where basidiocarps or fructifications, they are somewhat fleshy, they have a stipe or stalked, or he has also extended the concept to woody and sessile fructifications. Later on, the concept it evolved with the contribution from different mycologists, and Fries in 1821 he proposed a tribe polypori for these fungi, and from that gradually the concept of family polyperaceae evolved, and this family it included most of the polypores, but it excluded those genera where we have sort of lamellate pores. For example, we have lenzites as one of the genus where there is a formation of somewhat elongated lamellae-like pores they are formed. So they were excluded, all other they were put on in family Polyperaceae. Now later on, Polyperaceae, this term, it was the family was studied, molecular systematics, it gradually ventured into the field of taxonomy, and after that this polyperacy has now been split its members, they have been now distributed under 17 families and 7 orders of agaricomycetes. Now what we have been doing over the past few years, that is the practice of morphological studies. Of course, with time we have ventured into the addition of molecular studies also to supplement the morphological studies. So morphology in this case has also always been an important character to study, and we can have both macromorphological as well as micromorphological details which can be of significant importance while going for the taxonomy or identification of these fungi. If you look at the agarics, which you have studied in the morning, which you have been going through that feast of agaric world, in case of agarics, the basidiocarp development, at least in part, that takes place in soil. And during the last stage, there is a tremendous expansion, and these fructifications they appear. That happens in case of lamellate mushrooms or agarics. However, if you compare the polypores, in the polypores, basidiocarp development is always preceded by a visible vegetative stage. There appears some sort of vegetative growth on the wooden substrate before a fructification is formed in case of these polypores. And besides this vegetative phase, there is a very vast hyphal network that is developed inside the substrate. And eventually, this network of hyphae, it will emerge onto the surface of the substrate, from where ultimately mature basidiocarps, poroid basidiocarps, they develop. In some cases, we may have a sterile knob-like stage, for example, in some members of Ganoderma, and eventually that knob will develop and will form the fructification. Distinct asexual stages, they are rather rare among polypores. In most of the cases, asexual stages, they are not reported. But in some cases, for example, few members of family Hymenocetaceae, particularly there is genus Inonotus, there few species they have the asexual stages which are associated with the fructifications also. So that is of relevance. So now we will take up one by one, gradually and slowly, the different types of features which can be of importance while describing or identifying or working on these fungi. Now, most of the polypores, if we take into account the basidiocarp, most of the polypores, they have very stable and distinct shape of the fructification. They have very distinct and stable pattern of attachment of the fructification, and they are quite distinct in their color also. And the fructifications in polypores, they are somewhat tough, rigid, leathery, so they do not easily decay. Even if you collect them from the field, you can keep them for good number of days. And if you have dried them properly, they can be beautifully maintained in your herbaria. And you can make an independent section of these wood rotting fungi by simply drying and preserving the basidiocarp specimens of polyporoid fungi. So their shape, their attachment, their color, that is very, very stable and distinct. Some of them, they are now being exploited for the natural colors which are present in these fructifications. Let us take the common types. The range varies from rhizopinate to pileate. Rhizopinate means closely attached to the wood. Pileate means where the fructification forms an angle to the wooden substrate. Now, pileate basidiocarps, where pileae they do develop, they may be either sessile or stipitate in their nature. They may be broadly attached to the substratum, or they may have a distinct stipe or stalk which is visible in this case. So all these features, they must be carefully noted in the field while you make your collections right in the forest or any wooden substrate. Now, sessile features, the sessile fructifications, they may be broadly sessile, or in some cases, the posterior portion of the fructification, it may taper somewhat. So there may be gradual narrowing of the fructification that may also be there while attachment is being made. This is with reference to the sessile basidiocarps. Now, the stipitate basidiocarps, they may be centrally stipitate or laterally stipitate. So, position of this type, that is very, very important, whether it is central, whether it is lateral. If you look at genus Ganoderma, which is very, very important group of wood rotting fungi, their position of this type has been used now as a taxonomic character. You have to take into account the position of the stipe. Then the shape of the fructifications, it is highly variable. It may be drooping just like a pendate. It may be spatulate with somewhat spoon-shaped appearance, ungulate, broader from the front, gradually narrowing down. It can be triangular in appearance, that is trichoterous. It may be umbonate, somewhat swollen. It may be aplanate, where there is a broad attachment. So all these features, all these terms, they are regularly used while describing the basidiocarps of polypore or poroid fungi. So now you can see the range of variation in this slide. Here you can see this fellow is trying to separate a sort of this particular nature we refer to as rhizopinate. The fungus is growing unilaterally in one direction only along the substrate. This feature is called as rhizopinate. These are called as rhizopinate fructifications. Gradually, what happens in some cases, from margins, some angle that is formed. These are called as eresupinate effused reflexed. So there is a formation of some sort of reflexed portions. They are reflexed fructifications. And finally, you have the formation of these very prominent pilei. So these all, they form pileate group of fungi. So, and you can see and you can have a look onto this. So take to it. So you can see these all, they are broadly attached. They are sessile fructifications. Their point of attachment is broad to the substratum. And here in these cases, we will take on later on. Here, the attachment is there is some sort of narrowly attached pilei. They are there. And in Ganoderma, we will see later on, there is a formation of unique stipes. So resupinate fructifications, effused reflexed fructifications, pileate fructifications with broad attachment, and finally you have stipitate fructifications. Another feature to look onto this slide is the type of variation in their color. So you can see the color is very stable. For example, here you have typical white, creamish white to brown. Then here you have typical golden brown. These fructifications, typical golden brown fructifications, they are the characteristic feature of family Hymenocataceae. And that too, if you put a drop of potassium hydroxide solution, a drop onto this fructification, that portion will turn black in color. It's a unique xanthochroic reaction that takes place, which confirms that this is a member of family Hymenocataceae. This is a species of genus Pycnoporus. Pycnoporus, this sanguinis, where you have typical red colored fructifications. So color is very stable feature in these fructification. Here you can see the unique pattern of attachment of Ganoderma aplanatum. Broad attachment to the substratum. This is a species of genus Phalinus with imbricate type of fructifications growing in groups one above the other. So this is how there is variation with reference to the shape, with reference to the color, with reference to the nature of fructification, which is a stable character in these. Yes, we will proceed further. So you can see how they are growing in these groups. This is a beautiful member of genus Inonotus, which we have clicked while we were visiting a place near Dharamshala and McLeodganj. And this is the annual type of the fructification. Now, this is a typical species of genus Phalinus with perennial basidiocarps. In this case, every year there is a formation of, now this upper surface is called as abhymeneal surface. This lower surface is called as hymeneal surface or hymenophore. So this is the abhymeneal surface. This is the hymeneal surface. Every year in perennial species, a new hymenium layer develops in this zone. And when you cut a cross section, you will see large different number of layers of the tubes that is formed within one fructification. From that number of tube layers, you can make out whether the species is annual or it is perennial. That will determine the nature of fructification in these polypores. Similarly, here you can see unique brown color, non-laccate type of the abhymeneal surface, and this white colored lower hymeneal surface. This is the surface where pores are formed. This is the surface from where discharge of basidiocarps that takes place. So we have a hymeneal surface and we have an abhymeneal surface. Whenever you go in the field for collection of these fungi, always Remember to carry a good camera and try to click the close-up photographs along with their association with the host, taking by placing some reference in the form of a scale or any other structure. By this, you will do one thing: you will preserve the characters of fresh Basidio carp. And while you are clicking the photographs, try to click the photograph without flash of your camera, because when you use that flash, the natural colors of these Basidio carps, they are disturbed. So if you have good camera with good exposure, you can maintain these fresh Basidio carp photographs. And quality journals, they always welcome the manuscripts with all such good quality photographs, which give you an idea about the fructification. Another photograph which further gives you an idea about the kind of basidio carbs. So this is again a perennial fructification where gradually every year here new layer of hymenium that develops, and upper surfaces you can see it is very thick, leathery. This belongs to the genus Fomes in case of family Polyperaceae. So these are annual fructifications where you can see the formation of this sort of brown colored fructification. So color is important, the nature is important, and the shape, that is also very, very important and significantly used in the key characters. The second character, macromorphological character, that is with reference to consistency. Now, hyphal structure, they are basically responsible for the typical consistency of the fructifications. We have just seen that we have perennial and annual fructifications. Now, this usually in case of perennial fructifications, the fruit bodies are normally woody in appearance. And sometimes it is very difficult to remove these woody fructifications from their substratum. Now, this again depends upon the type of hyphal structure. Now, students and other friends, we all are aware that we have the basidiocarp. We have in basidiomycota, we have the formation of basidiospores. These basidiospores are discharged, released from the basidium. These basidiospores, they germinate, and the first hyphae which they form, they are called as generative hyphae. And if in a fructification we have only generative hyphae, the hyphal system is called as monomitic. Gradually, in different fungi, from generative hyphae, there is a formation of different structures. Generative hyphae can form skeletal hyphae. They also form or give birth to binding hyphae. So, principally speaking, in fungi, we have three types of the hyphae: the generative hyphae, which are usually thin-walled, first formed after the germination of basidiospore. Then we have very thick walled, narrow lumened skeletal hyphae. Those are usually unbranched, without septa. Then we have irregularly branched, thick walled, narrowly lumened structure called as binding hyphae. So you have generative hyphae, skeletal hyphae, and binding hyphae. Depending upon the type of hyphae present in a fructification, we can call as hyphal system to be of three types. And determination of hyphal system in Basidiomycetes wood rotting fungi is very, very important for applying any taxonomic key for their identification. If you have only generative hyphae, they are present in the fructification, the hyphal system is monomitic. Such fructifications which have monomitic hyphal system, they are usually soft, and they may shrink considerably when they are dried. And in these fructifications, there is also possibility of change of color. So those changes we need to note down very carefully. What is the color when it is fresh, and what is the color change after drying? You have to note down both of the abhymeneal surface as well as of the hymeneal surface, monometics. If in addition to generative hyphae, if we have either this binding hyphae or skeletal hyphae, generative hyphae will always be there. In addition to generative hyphae, if we have either skeletal or binding, it will be dimetic hyphal system. And if all the three types of hyphae are present, we call that as trimetic hyphal system. And hyphal system concept, it is very, very important and very relevant while going for the taxonomic studies on wood rotting fungi, particularly these polypores. So trimetic hyphal system bearing fructifications, they are very tough, they are very difficult to tear apart, and they give the fructifications typical woody or leathery appearance in case of these fungi. Then in some cases, we may have an aroma or taste. Polypores usually they have no distinct taste or any sort of fragrance except for few. There is one such example of Tramates suaveolens, where you have typical anise seed-like fragrance that is emitted by the young fructifications. This is what Tramates suaveolens it looks like when you see its fructifications. Edibility is usually polypores are not edible because of their leathery nature, but some of them they have the edibility. We have Latiporus sulphureus, which is the wood chicken, highly delicious, highly nutritious. We have Graphiola frondosa and we have Fistulina hepatica. Few of them they are edible. Usually these characters they are not used regularly while applying the taxonomic keys for identification of wood rotting fungi. So with this background of the pileus in mind, let's move on to the pilear surface. So we have to see the surface of the pileus carefully just before compiling our data. So you have made a collection, you have noted down the nature of rectification, annual or perennial, you have noted down its shape, you have noted down its mode of attachment, you have noted down its color, both of upper surface as well as lower surface. Besides color, we have other details to be noted. In many species, pileus is having distinct color which remains reasonably unchanged during lifespan, more precisely in perennial fructifications. In other cases, the color may fade because of the destruction of pigments due to weathering that may take place. Light and white colored species, they often become darker because upper layer of hyphae it gradually collapses and makes the fructification darker in appearance. So these changes we have to note down. What are the changes, what are the features which are present in fresh condition, and if there is any change happening with age, we have to note that change carefully. Then the pilear surface. Is it glabrous, that is smooth, or it is covered with some sort of hair? This is a character which should be carefully noted down, which is carefully noted down while making the macromorphological account. The glabrous surface, if it is without any growth, it can be dull or shiny. This is character very, very importantly used in Ganoderma. It may, when you cut a simple section, a small portion is cut with a simple blade, whether there is presence of a layer which we call as refer, this which we refer to as cuticle, or with some zonation, some radial lines, or some raised ridges. So these fresh features, they must be carefully noted down before having the details, before compiling the details of the fructification. This is with reference to glabrous surfaces. And second option is presence of hair. Now, here, if they are present, they may be persistent or they may erode with age. Young fructifications may be hairy, and mature fructifications, they may be devoid of such hair. Hairs, they often tend to occur in these zones. This feature is again of importance. And the perennial species, they may develop certain cracks with age. So that feature is also to be carefully noticed. So now you can see the four typical examples where we have different types of appearance. This is a typical upper or pilear surface which is having these hair, and hair are arranged in the form of zones. So you can see the hairy or tomentose nature of the pilear surface. Then you can also observe there is change in color. This is the marginal color. So at margins, the color is somewhat different. Rest of the fructification is differently colored. This complete range of color, that is from golden brown to brown, that has to be carefully observed. In these cases, we don't have hair. They are all glabrous. But here you can see the formation of some sort of zones of two colors. The outer zone, that is formed of light brown color, and the inner zone is somewhat dark brown or blackish brown. So this type of variation of the pilear surface, the main body and the margins, that must be carefully observed. Now take this example of hymenocetoid, this mushroom, this polypore. You can see towards the periphery, we further have formation of alternate light and dark colors. And margins, they become creamish white in color. So they all such details, color variations, nature, presence or absence of hair, all these features, they are of extreme importance, and they are used while going for the macromorphological details. Margins, we have already mentioned. Margin and its development, that is very, very important in resupinate fructifications, where we have closely appressed growing fructifications. Their margins are very, very important. In these areas, sometimes we have the formation of hyphal cordons. Hyphal cordons, they may be associated with margins. This has to be noticed. Color of the margin on abhymeneal surface, we have already seen that variation. So that variation is an important taxonomic character. In case of stipitate members, stipe may also be differently colored. So color of the stipe has to be carefully noted. Shape of the stipe: cylindrical, subcylindrical, bulbous, clavate, subclavate. These are the principal shapes of the stipe which are there. And position, whether it is central or it is eccentric or it is lateral in position, that has to be carefully noticed. Sometimes a single stipe may give birth to more than one pileus. So this is again an important character which is used to distinguish few species within genus Ganoderma also. So pilei in these cases, they are usually fan shaped. So this is an important character which one has to notice down very carefully in case of these fungi. Then let's move on to the pore surface or the pilear surface. So if you look carefully at the lower surface, on the lower surface, what we will find? We will find these narrow openings. And these narrow openings, they are called as pores. So one has to carefully take the portion of basidiocarp, observe its poroid surface carefully, and you have to note down certain characters or features carefully. The first character is the pore size. Now the question arises how the pore size is measured. That is simply measured by placing a scale on the poroid surface. And most of the these polypore taxonomists, they use the character number of pores per millimeter. So you have to take a scale, place it on the hymenophore or poroid surface, and then count manually the number of pores per millimeter. And you have to take minimum 10 to 15 readings from margins towards the center of the fructification to give you a range of number of pores per millimeter. That is the first character. Second character, shape of the pore. That is also very, very important. So you have to note down what is the shape of the pore, whether we have regular pores or irregular pores. So sometimes in few cases, pores they tend to fuse and the hymenophore becomes somewhat irregular. So this character has to be noted down. Shape of the pore, whether it is polygonal, circular, or it is somewhat elongated. Here you can see the presence of polygonal pores, and presence of polygonal pores, that is the characteristic feature of genus Hexagonia. So that genus is easily identified in the field on the basis of unique polygonal pores. Then we have sort of these lamellate pores which are elongated, and they, that is the characteristic feature of genus Lenzites. So such characters, the shape of the pores, number of pores per millimeter, they are easily observed and they are significantly used while applying the taxonomic keys. Other than this, you have to note down the color. What is the color of the pore surface, both when fresh as well as on drying? And another important character is the margins. So margins should be carefully observed on the hymeneal surface also. And there is one important character, whether margin is fertile or sterile. If the margins are having pores, those margins are called as fertile margins. And if the margins are without pores, they are called as sterile margins. So if they are fertile or sterile, we must note that feature also. So these are some of the very, very relevant and important type of the macromorphological details which we must keep in our mind while noticing or observing these fructifications, either in field or in laboratory using a simple dissecting microscope or a binocular. Then we come to the micromorphological details. Methodology we will discuss later on, but when you take a portion of fructification, you can crush it, you can tease it, or you can make a cross section. And from those cross sections, we can study different micromorphological features. And to start with, the very first character that we have to look for is the type of hyphal system. Whether we have monometic hyphal system, dimetic hyphal system, or trimetic hyphal system, that will be evident when you make good microscopic preparations. If there are only these thin-walled septate, regularly septate hyphae, which are called as generative hyphae, the hyphal system is monometic. If in addition to these generative hyphae, we have these thick-walled, unbranched, aseptate skeletal hyphae or binding hyphae, they are called as dimetic. If all three are present, the hyphal system will be referred to as trimetic hyphal system. So first character which we need to observe or note is the type of hyphal system. Then we have to look for the few features which can be very unique in some genera. We have to look for the length of the hyphal cells. In some genera, we have short-celled hyphae. Before that, we have to look for the clamp connections. You must be aware about the presence of monokaryotic and dikaryotic hyphae, and in basidiomycota we have a prolonged dikaryotic phase. And clamp connection is one such mechanism which is responsible for bringing about the process of dikaryotization. This is one such mechanism. Others are, of course, there. So, in some members of Basidiomycota, when you observe these generative hyphae carefully under microscope, usually at 40x or 100x, you can see at the septa presence of these clamp connections. So, if clamp connections are present, hyphae are clamped. If they are absent, we call them as simple septate hyphae. Then you have to look for the length of the cells of these hyphae, whether they are short celled or they are normal. In some cases, just before the septa, the hyphae tend to swell. So there are swollen hyphal portions just before the septa. They are called as ampullate septa. Then hyphae may be smooth, that is without any deposition, or sometimes on the hyphae you can find certain granules or crystals. They are called as encrusted hyphae. Within family Hymenocetaceae, we have a Formitiporia genus that is distinguished from other genera on the basis of presence of encrusted hyphae. Other features they are studied later on, so you can distinguish on the basis of you can have a broader idea even on the basis of these features. Then in a fructification, we have certain ancillary or additional structures. These ancillary or additional structures, they are called as cystidia. They originate from hyphae. Their function is to strengthen the hymenium and to create distance or to separate the basidia so that basidiospores can be easily discharged. So these structures, they are called as cystidia. So they are visible in the hymenium. So you have to be very carefully prepare the microscopic preparations, and in addition to basidia, you have to look for these structures more precisely in the hymenium portion. These structures are called as cystidia. They may be long tubular cystidia, they without any encrustation. They may be encrusted cystidia with crystals being deposited throughout their length, or they may have crystals deposited at their tip. There are so many variations. Their shape is very much variable, or their extent of encrustation, that is also variable. So shape is to be taken into account. Presence or absence of clamp connection is to be taken into account. Presence or absence of crystalline deposits over these cystidia, that is another key character which is required for making these microscopic details to be used for taxonomic purposes. So this is how the cystidia they look like when you do their photomicrography. Encrusted cystidia with these deposits. Then we have these leptocystidia, which are having this sort of subclavate appearance. Then in some cases, cystidia, they contain some oily contents. These are called as gliocystidia. Some of the genera, they are very unique in having oil-filled cystidial elements called as gliocystidia. Then we do have this important structure called as the stephanocyst, where in the middle of the cystidia, there is presence of some teeth-like structures. Then we have asterocystidia, where at the tip of the cystidium, star-like deposition of the crystals that is done. Then we have leginocystidia, which have a broad base and a pointed tip, and at the tip there is presence of these crystalline depositions called as leginocystidia. We have ornatocystidia, where tubular cystidia, they have regularly placed crystals. And I must tell you, all these features, they are very well preserved and well observed in the microscopic preparations. Few more cystidia, you can see the presence of lyocystidia, which are thick walled, capitate cystidia, which have cap like endings, and you can see these asterocystidia, star like cystidia, which are thick walled. They are very much easily visible in the microscopic preparations. This is again the presence of clamped cystidia. Another word is pseudocystidia. Sometimes in dimetic or trimetic species, skeletal hyphae, they extend beyond the hymenium. So there is no septa formed in these extensions, and such structures are referred to as pseudocystidia. बेटे मेरा हो दे charger. Then the third important structure, that is the presence of uh, the spore producing structures, and these spore producing structures, they are called as basidia. So basidia, they are the structures which form the hymenium, and their shape is variable. Their basal structure is variable. So we have to take into account their shape, their position. First is position. We can have two possibilities: whether basidia are terminal, that is they are ending or they are forming the tip of the hypha, or plural, where hyphae, they are keep on running, and they give birth to many basidia. So we can have terminal or plural basidia. Then the shape is variable: tubular, somewhat constricted, stalked, where basal portion is stalk like, clavate, subclavate. So many different types of basidia, they are there on the basis of shape. Then in some basidia, oily or granular contents, they may be present. This is another important characteristic feature which is associated with taxonomy of wood rotting fungi. Number of sterigmata, that is also important. More commonly, we have four sterigmata per basidium. But rarely there can be two sterigmata per basidium, six or even eight sterigmata per basidium. So this is another important key feature. Then the length of the basidium, that is also observed in this case. Hyphae, in case of hyphae, what we usually take the width of the hyphae, range of width of the different hyphae that we observe or notice. So this is how the basidia look like in the microscopic preparations. You can see the tubular basidia. You can see somewhat these subclavate basidia, basidia with oily contents like this, and basidia with six sterigmata, four sterigmata, so on and so forth. All these features, they are beautifully available, easily observed under microscope, simple light microscope. Then we have the basidiospores. The shape of the basidiospores, that is highly variable. We can have ellipsoid, broadly ellipsoid, subglobose, globose, cylindrical, or navicular type of the shape of the basidiospores. Their wall thickness can be variable, thin walled or thick walled, that can be there, or they may have some spines called as verrucose basidiospores. Sometimes we have broader depositions referred to as tuberculate basidiospores. Some pattern like observations that can also be associated, which are called as reticulate basidiospores. So all these features, they are carefully observed. Now there is another term written over here, repetitive. In some cases, a basidiospore germinates and it again gives birth to another basidiospore directly. So this repetitive basidiospore character is also important. So you can see the range of variation in shape, the range of variation in the wall, and that is clearly visible in this slide. Different range that is associated. Globo, globose, subglobose, cylindrical, subcylindrical, smooth, or spiny basidiospores. All these characters, they have to be carefully observed. You can see sometimes even from the even from the shape of the basidiospore, one can notice or get an idea about the generic identification. For example, in case of genus Ganoderma, we have a truncate type of the basidiospores. They are not shown here, but I'll show you later on in other projections. So all these microscopic characters, they can be carefully observed by taking these preparations, and that can yield you significant data. And ultimately, you can see how the construction of the basidiocarp is there. So if you take a portion of basidiocarp, you with a simple blade, you just cut a cross section of the hymeneal surface, and you will see this is how a pore looks like. This is the opening or pore. This is the hymeneal portion where we have basidia, and this portion, this region is called as the trama region. So these are two adjoining pores with gradule with their hymenium on both sides. This is how the structure is maintained. This is with reference to the resupinate members. There you can see this type of appearance of the hymenium. So all these characters, they are observed carefully. They are noted down in the form of a description. Then we compare them with the published literature and they can be identified. We have explored different parts of Himachal, Punjab, Uttarakhand, some portion of Jammu division, and on those bases, we have made certain studies. These are reported in all sort of forests which we have come across. You can see mixed coniferous forests. You can see the deciduous forest dominated with quercus species. The beauty of this work, taxonomic work, lies in that you get a chance to visit new and new places, which otherwise is not easily manageable. But this happens in such studies. Beautiful landscapes, you can see all these forests, they have good growth, good diversity of these wood rotting polypores. So sometimes you come across so many interesting features. Churdhar, that is most diverse range. Chopal is there. Chakrata. So all these places, they have been visited by our team for the collection of these fungi. So we have visited all these localities ranging from Punjab to Jammu Kashmir and other portions. So this is how these fungi they grow. They usually occupy such logs, stumps, or decaying wooden substrate. And let's come to the methodology. So these fungi, since they grow in close association with the wooden substrate, they have to be carefully removed from their substrate so as to collect them in good condition. So we need certain tools. We need a hammer and a chisel. With this hammer and chisel, the fructification can be easily separated along with the portion of wood. In some cases, even a simple knife is sufficient to take out the basidiocarp along with the portion of wood. Then whatever characters we have mentioned, they are observed under preliminary examination. The association, name of the host, name of the locality, type of the forest, then the nature of the basidiocarp, its color and its shape. They are all observed, they are all noted down in a field notebook. Then we go for, this is how the collections they have been made. These all are teammates, they are collecting these fructification. Now these fungi, they usually prefer to grow on shaded parts. They very rarely they grow on the exposed portions. Usually they grow in these shaded parts. This was my first collection trip when I joined my PhD way back in the year 2003, and that is very first experience which comes into mind. Then we do these photography that needs to be done, and we make these preliminary examinations. Then we need to get a spore print. Spore print is basically an impression of spores on a glass slide. So what is done? A small piece of a chart paper is placed. On that, a clean glass slide is placed. And on this clean glass slide, a portion of the fructification is placed with the hymeneal surface facing the glass slide. And this is left for overnight. There is a forcible discharge of the basidiospores from this fructification, and ultimately the impression comes on the glass slide that is covered with another glass slide, and ultimately it is packed. So all the features of the basidiospores, they are observed from this spore print. The specimens, they can be dried either in sun or they may be dried using electric dryers. Then comes the microscopic observational part. Now, what we do, we have two types of the approaches. First, we can cut cross sections of the fructification part. So fructification is, its cross section is cut. That is first studied in KOH solution, 3%, 5%, and 10%. So there we observe different features: hyphae, basidia, cystidia, and basidiospores. Then we put this stain Congo red. This is 1% prepared in distilled water. Congo red stains the wall of the fungal structures, and boundary becomes very, very clear. This gives you a clear idea about the shape of the fungal structure. Then to study the contents, if any, we use floxin, which is again 1% in distilled water. Then we study two more characters. One is a cyanophilus reaction, other is amylase reaction. So we for studying cyanophilus reaction, we use cotton blue, usually 1% in lactophenol. So if the wall of any fungal structure turns blue or bluish black in cotton blue, the structure is said to be cyanophilus. Similarly, if the wall turns blue or bluish black in Melzer's reagent, the structure is called amylase. These two reactions, they are very important with reference to basidiospores. Amylidity and cyanophilus, these two features, they are used in many of the keys. Besides these features, we note down the size range of all structures. We note down the width of the hyphae with the help of camera lucida. We note down the size of basidiospores, length and breadth, basidia, cystidia, all these characters. And for each character, minimum you have to take 20 observations, only then you get a range of the size range of all these microscopic structures, that is very much required for confirming the identity of the structures. And ultimately, with the help of very good sophisticated microscopes, you can get the photomicrographs of key characters of these genera. The macro and morphological details, they are documented in the form of description, and they are compared with the literature, and then you can reach at certain conclusion towards the identification. So these are some of the standard monographs which are available for publication. Now, using this, we have explored the Agaricomycetes wood rotting polypores of northwest India, and we have described a good number of species. So, this is how a standard description is based upon. So, you take the details of abhymeneal surface, hymeneal surface, compile the microscopic characters in the form of line diagrams, supplement it with photomicrographs, then you identify it, compare with literature, and you can comment on the status of your species. For example, this is Nomenscularia canherei, which is the first record for India. We have published a new species of Phalinus, that is a Phalinus himalaicus, on the basis of this description which we collected from a part of Himachal Pradesh. Similarly, we have published Phalinus indicus, very unique species on the basis of its unique features which are somewhat close to raw berberriness, but it is very. Now, this is typical resupinate fruit body. This is how a resupinate fructification looks like. Similarly, we have more species of Phalinus described as new. We have Porphyloporia, unique, narrowly attached fructification. Then we have this another resupinate member, Oxyporus obducens, which is a new record for India. Then we had these Entrodia species. You can also extend your studies to certain applied aspects. We can also explore the histopathological details. What is the level of penetration of the hyphae in the substrate, intracellular, intercellular? So that can also be studied by cutting TLS or RLS of the wood and tracing the hyphal penetrations. Ganoderma, we have worked and we have about 28 different species of Ganoderma. This is what we call as ganodermoid spores. So exclusively from the basidiospore character, one can have the idea of the genus. Now, these are the two variations you can see in their surface. This is shiny, lacquate surface. This is dull, that is called as non-lacquate surface. And the two species, they differ from each other in many features. These are typical stipitate members having a stipe and a pileus in genus Ganoderma. You can also supplement your studies with the help of scanning electron micrographs of different important structure. For example, here we have included the SEM images of spores of Ganoderma. It is Ganoderma lobatum, which is a new record for Punjab. You can see its beautiful spores. Similarly, few more species of Ganoderma. Here you can see branched stipe, that is characteristic feature of Ganoderma ramosissimum. Still, we have Racinium, Ganoderma subumbraculum. All they have been compared on the basis of their micro and microscopic details. This is another species which we published in Mycotaxon in 2015, a species of Entrodiella, that is Entrodiella indica. All such details, they have been published. So all this work would not have been possible without the contribution of two very important in my people in my life. He is Professor Gurpal Singh Dhingra, who is my mentor, my PhD guide, and still guiding me a lot. Second one is Professor N.S. Attri. He has been promoting me like anything, and he has been of great help right from the word go. And I wish to thank and acknowledge the contribution of both these mycologists in this little bit of discussion. I'm also thankful to these three fellows. He is Professor Nils Hallenberg from University of Gothenburg. He is Professor Leif Riverden, and he is Professor Ewald Langer from Kassel University, Germany, who have been of great help in providing literature, confirming the identifications, and I'm equally indebted to DST and UGC for financial assistance. And all this would not have been possible without these research scholars, and that too majority of the girls who never hesitate in going to the field making collections, and five of them, they have submitted their PhDs. These three have already been awarded. Her PhD reports, they have been opened recently, and this girl, she has submitted her thesis 2 days earlier, on right on Friday. These two are currently, they are continuing with their polypore studies. So I am extremely thankful for such a dedicated team. So at the outset, I am also thankful to the organizers, and also to the Mumbai unit of Mycological Society of India, Dr. Deshmukh and Dr. Shashi Rekha, and the entire team who are making great efforts for popularizing mycology. So that's all from my side. And if there are any queries, questions, it will be an honor to answer. Thank you very much. Hope I have not taken much of time, and it's as per the time allotted.
Thank you. Thank you very much, Dr. Avneet Singh, sir. It was very informative talk, and must be enriching for all the students and teachers, researchers working in the field of mycology. Before proceeding towards the presidential remarks, we have a few queries with us from the students.
Yes, please, sir.
This is the query. Actually, you have addressed the same in your talk, but I think you can focus more on this issue. This is the question raised by Tepin Rakhe and Karabi Devi.
Yeah.
Sir, which literature can I follow for identification of polypores?
Yeah.
I would have
Yes. I wish
second
to.
question, which is just attached to this from the Tepin. We should use hypal system for identification from the culture or from the fruiting bodies, or both.
Yes. So these two are very valid points, because we must know the standard literature. As far as literature is concerned, I wish you all to have the volumes. Try to procure the volumes of polypore, genera of polypores, which have been composed by Leaf Riverden in the form of 6 volumes. So genera of East Asian polypores, genera of European polypores, poroid fungi of Europe. These are some to mention which have been published by Leaf Riverden. And from Indian perspective, I would like to mention you, which I forgot, I should have mentioned, the great contribution by Professor J.R. Sharma from BSI Dehradun. He has published 3 beautiful publications. One is Hymenocracy of India, that was published in 1985. And there is another publication that appeared in 2012, Aphilophrails of Himalaya, where he has beautifully summarized the work done from Indian perspective. You can follow any of these, and if anyone from this side is in need of literature, just give me an email. We have in our lab all this literature available. We can send you the xeroxed copies, or you can visit our place whenever you feel like situation is good to visit, and we will be happy to share. As far as hyphal system is concerned, the third approach, that is both from fructification as well as from culture, that should be an ideal approach to comment on the hyphal system. But the literature which I have just mentioned, majority of the keys, they stress on the hyphal system, which is worked from the fructifications.
Thank you, sir.
Thank you.
One more question from LKV Pawar.
Yes, sir.
And he's asking that are there any polypores fungi used for medicinal therapeutic purpose for any disease as we are using the plants?
Yes, sir. There are good number of examples. If we work on the literature, if you survey it carefully, we have Ganoderma lucidum, which has been called as Rishi mushroom, and lot of work has been done, and even some of the products they have been released for commercial utilization. Besides this, we have Trametes versicolor, which was earlier called as Coriolus versicolor. From that, good number of products they are now available in market, which also have anti-cancerous or anti-tumor activities. And we have a genus called as Inonotus. The species of Inonotus, they have been exploited for many these serious ailments of human beings, particularly in China and Middle East. This is a new area to venture in. We have started these studies also. We have taken initially with the antioxidant studies, and the results they are quite encouraging, and we are planning to further take those studies, and we have now initiated an exclusive work on anti-cancerous properties of some of these polypores.
Thank, Thanks a lot, sir.
Thank you.
Actually, your talk was so self-explanatory that there must be clear-cut guideline for all the students, and it was guideline for all the students to carry out their research in the further course of action.
Yeah.
And we can see the huge appreciation for all the students and teachers and researchers in the YouTube chat box. That was
Yes,
the
sir.
appreciation for your contribution in the field of mycology, and that is the acknowledgement for your talk, which you have delivered so lucidly in this session. Thank you very much, Dr. Avneet Pal Singh, sir.
And
Now I extend my request to the Honorable Chairman of this session, Dr. Yashpal Sharma, sir, for his presidential remarks.
Yeah, good afternoon, everyone.
afternoon, sir.
So, I guess, am I audible?
Yes, yes, sir.
Okay. So, I guess that it has been a wonderful, wonderful session. And I won't stop here by just saying that it has been a wonderful session only, because there are few observations which I was keenly making, that it was appearing as if Dr. Avneet has us taken all of us virtually into a forest or into a field. And this is the beauty of the workshop lectures, wherein everybody's glued to the screen, not to miss any statement, not to miss any word, not to miss any kind of information which is very, very meticulously, presented by the learned speaker. It was interesting to understand many things related to systematics and diversity of polypores, and particularly when he was referring to northwestern part of Himalaya, which is, biodiversity wise, it is very, very rich. There are several things which we certainly need to understand about all this northwestern part of Himalaya. One of the things is that since it has got a lot of diversity with respect to flora, so therefore fungi are, they have availed that opportunity very well. In fact, they got a lot of flora to flourish over there, and in the process, they also got benefited. Dr. Avneet very nicely, divided his lecture into two components, and the first component was really, really informative for the youngsters who actually are into the field, or who wish to take up this mycology, because we have been hearing about the concern shown by the top mycologists, the senior mycologists, that this is one such field which is remaining orphaned, or this unfortunately is not given so much of priority in all kind of diversity studies, whether you talk of people biodiversity registers, or they all are looking at those things which are very fascinating to them. But I guess this one such group of fungi in general, and particularly the macro fungi, including the fleshy as well as these polypores, they certainly are the organisms to reckon with, and we all need to be very particular. So that was the best part, wherein he has very intelligently dealt with all the kind of characters and all the kind of techniques which are to be employed to reach at certain conclusions. Well, starting right from the macro morphological characters and taking us through the realms of the micro morphology. That was the best woven, concept, which was very well presented by Dr. Avneet. For that, he needs a lot of appreciation and applause from all of us. Um, he also touched upon evolutionary part of that, how those gymnocarpic through hemiangiocarpic kind of basidiocarps, uh, they gave rise to then ultimately to the angiocarp, or the latest we see that they are the advanced kind of the protein bodies. And then uh, history part, that was really wonderful. And then very nicely when he touched about the hyphal systems, the spores, the basidia, and the ancillary structures, particularly the cystidia, I was very happy because I think a couple of years ago when we were at Pune, so there was a lecture presented by uh, Bart Baik. So what he said, that this fungus, every fungus is, it has got hidden identification. You need to actually venture into it. You need to go into it. You need to expose that, where that jadoo ka identification is hiding. And the fungus has kept every character inside it, and which makes it quite different from others. So that small needle you have to look for in pile of hay and all that. So that's the beauty of things, that the ancillary structures, as the you know the reproductive structures are concerned, they of course are very important, but the ancillary structures or the sterile structures, they all are equally contributing. In fact, in certain cases, they are giving you the indication that, oh, my name is this one, look for me. So that was nicely done, and every statement of his was being corroborated with the pictures, the drawings, which have been very nicely presented. Making camera lucida drawing is really a cumbersome job, though we are now having advanced techniques, but just look at the amount of hard work they have put in. So overall, it was a very well understanding, and I'm sure with the kind of attention given by, or with the kind of response, I came to know that many young students are adhering to such lectures through YouTube and other channels of sites of the Shivaji College. It's really kudos to the speaker, and he certainly needs applause for this. Few things I wish to tell about Professor Dr. Avneet. Of course, he will be professor very soon. The company, the kind of commitment, the kind of the patience, the kind of zeal he develops, and the zeal he puts in his students, and of course utmost respect for the teachers. So he has been very lucky to be at a center wherein you had the stalwarts working, and he has picked all thread very very nicely and very very keenly. And of course the patronage of his own guru, his teachers around, Professor Atri being there, Dr. Manuruchi being there, and then the Patiala has been known for the history with respect to the macrofungi. I could see their herbarium and all that. But the support again, apart from these, our own teachers, the Riverdhan association with Riverdhan, I can well, he would agree that he's the father of polypores, I would say. And then Hellenberg, and then with the Langer. So such kind of associations, they make you to understand all intricacies of such diverse fields. But only thing is that this doesn't come of its own. You have to get into such kind of ocean to get that amrita, that kind of knowledge. So you have to extract that. So I'm really very very happy. So Dr. Avneet, heartiest congratulations to you. And having said this, I also wish to thank Dr. Deshmukh, who who very, he was kind enough to give me this opportunity to interact with all the mycologists, those who have joined directly, indirectly for this workshop. So thank you, Deshmukh sir, for your kindness. And of course, I was blessed to have the presence, august presence of a lot of good people from whom we have been learning: Professor Lakhanpal, Professor Manorachari, Dr. Atri, Professor Atri. Then I could also listen to Professor Surya Narayanan, Dr. Sanjay, and the teamwork shown by Dr. Hande. And I was referring to that a very well knit, organized workshop. And I'm sure that one and all would have got a lot of information, would have been benefited from this. So I wish that such kind. This is from here we can take the lead. We can also, others, those who are watching, their institutions can also take a lead, and they can organize similar kind of workshops on other areas, frontier areas. uh, I have many things to say, but I think we have to move together. There's always a time constraint, and I again congratulate the Shri Shivaji Science College, and of course the Sant Garge Baba Amravati University. I'm very fond of visiting that place. I've been going to the refresher course HRDC college. So I have special memories associated with the Maharashtra in general, and of course Amravati in particular. So our heartiest wishes to everyone, and thank you once again, everyone, for providing me this opportunity. Thanks.
Thanks a lot, sir. What a wonderful remarks you placed for the very lucid talk delivered by Avneet Pal Singh, sir. And it was indeed the pleasure to hear you on the same line of thought as Avneet Pal Singh, sir, have projected. I think this was the best combination of the speaker and the chairman for the session. And whatever is the combination, that combination was reflected from the talk and the presidential remarks for the session. Thank you very much, sir. At the end of this technical session, I must place on the record the acknowledgements to the chairman of the session, Dr. Yashpal Sharma, sir, the guest speaker, Dr. Avneet Pal Singh, sir. Then I must acknowledge the huge contribution by the MSI president for the Mumbai unit, Dr. Deshmukh, sir. Similarly, Dr. Shashi Rekha ma'am is also with us. I must acknowledge her presence in
Yes.
the session. Atri sir is also with us, and all the teachers, researchers, and the students who are there in the YouTube platform. I thank all the participants, all the resource persons and chairmen of different sessions. And with the permission of the chair, I declare that fourth session of the workshop is concluded. Please give us a few minutes to arrange the decorum of the stage. We'll just start with the valedictory session because valedictory chief guest is with us. We'll start within 2 or 3 minutes. Please give us 2 or 3 minutes. We'll certainly start in time. Thank you. Thank you very much, sir.
Thank you.
Thank you, sir.
Thank you. Thank you, sir.
Thank you, Avneet sir.
Thank you very much, sir.
Thank you, Yashpal sir. Thank you.
Sachka, sir.
Always welcome, everyone. Always welcome. Thank you. Pleasure.
Thanks to all of you.
Hello, everyone. We are here in the final phase of the National Online Workshop on Fungal Systematics and the Technological Advances organized by Sri Shivaji Science College, Amravati, Department of Botany, and Mycological Society of India, Mumbai unit. So I humbly welcome all the dignitaries on the dais. Today, we are having with us the Honorable Chairman of the session, Dr. H.S. Lunge, sir, who is the IQAC coordinator for our college. We are blessed with the presence of our chief guest, Dr. J.A. Tidke, sir, who is Professor and Head Department of Botany, Sant Gadge Baba, Amravati University, Amravati. We have with us Head Department of Botany, Professor B.K. Dorkar, sir, on the dais. With him, two dynamic organizing secretaries, Dr. D.V. Hande, sir, and Dr. Ganesh Hedau, sir, is also occupying the chairs on the dais. I extend warm welcome to all the dignitaries on the dais. Similarly, I must welcome all the scientists, teaching faculties, researchers, and students who have joined the meeting in the Zoom platform and the YouTube platform. So before starting the valedictory session, I must say something regarding the entire chronicle of events happened throughout the time when we have inception of the workshop. Actually, we all were in the predicament situation of the online teaching, online classes. It was like for the teachers and the students, it was like a caged classroom. And to bring all the students and teachers out of that caged classroom, we were thinking to plan some exotic activities through which we can bring all the students out of that frame. And we have formulated the proposal of this one national workshop, and that was labeled as the national workshop on the fungal systematics and the technological advances. Why we have chosen this theme? Because this is the department which was having the huge, long history of the subject studies and the research in mycology. This is the department which was recognized for the first time by then Nagpur University as the research lab, and many of the teaching faculties devoted their contributions in the research in mycology. And that's why this is one of the pioneer department in the study of mycology. And that's why we have some sort of authority to organize such kind of event with us. So we have planned the national workshop, and very importantly, this was the workshop in collaboration with the Mycological Society of India. Once we have announced this workshop for the national students, teachers, and scientists, it was surprising. It was overwhelming response throughout the India and abroad, and we have received huge number of participants for the workshop. It was the boost for us, and eventually we have meticulously planned the entire workshop and come up with this 2 days feast for all the students and teachers. My dear friends, this was the workshop which was planned by our dynamic leaders, organizing secretaries, Dr. D.V. Hande and Dr. G.B. Hedau, sir. I must invite Dr. D.V. Hande, sir, to summarize the entire workshop through his summarizing remarks on this platform. Please, sir.
Good afternoon, one and all. Dignitaries on the dais, Chairman of the today's function, Professor H.S. Lunge, Coordinator IQAC, Sri Shivaji Science College, Amravati. Chief guest of today's valedictory function, Professor J. Tidke, former Vice Chancellor, Santa Gadge Baba, Amravati University, Amravati. Head of the department, Professor B.K. Dorkar. My colleague, Dr. Ganesh, and entire team of the botany, Dr. Dinesh, Dr. Rekha Magirwar, Dr. Tushar, Dr. Swati, and Avinash. And the respected luminaries, esteemed MSI office bearer, Mumbai unit, members of other organization, chairman of various sessions, special invitee, and dear student from various part of the India and abroad also. Those are also on YouTube platform. I have great pleasure to sum up this event, 2 days national online workshop on fungal systematics and technological advances. We have planned this workshop with more valuable and applied concept in the field of mycology with reference to fungal systematics and technological approaches. It also aims to inculcate and foster research culture in mycology and original thinking as essential in the age of openness and globalization for knowledge-based society. The workshop was also aimed to motivate students to undertake mycological basic as well as applied research. We have been listening to eminent speakers deliberating in the area of mycology since last 2 days. And all these valuable insight gave us new perspective for the research domain in the field of mycology. As soon as this opportunity knocked our door, we tried to communicate the announcement with the participant through all online tools to the institution through Google Forms, Telegrams, WhatsApp, email, social media, and also personal level. We have received a remarkable response for this workshop, which has boosted our morale and encouraged us to organize similar event in future also. The workshop was open for all, wherein many senior mycologists, professor, industry person, UG student, PG, PhD student from India and abroad registered and participated. More than 1,100 researchers have been registered for this national workshop. Let me brief about this today event. I must mention here that when our college decided to organize the workshop on topic related to mycology, the option to collaborate this event with leading prestigious organization that strike us was Mycological Society of India and Mumbai unit. Thanks to Dr. Deshmukh sir, Dr. Shashi Rekha and Dr. Mahavir Goswami for accepting our collaboration. It's our privilege and good fortune that this eminent society accepted our proposal and joined us within the journey towards knowledge and enlightenment. I am thankful to all office bearer of the society. On the first day of the inaugural session, in the introductory speech, Dr. S.K. Deshmukh focused on the different activities of the MSI and Mumbai unit specially organized during the COVID-19 pandemic, like organization of mycology webinar series. Professor M.K. Rai, who is the world-renowned mycologist, in his inaugural address has congratulated the organizing team and their contribution for the subject, particularly in attracting students toward the mycology. He has pointed out that there is a need to conserve this branch and research must be oriented from basic to applied level. As we are losing taxonomy at alarming rate, he has mentioned that there should be one mycologist and one fungus correlation for the detailed studies. Honorable Principal Dr. V.G. Thakre, in his presidential address, appreciated the efforts of MSI Mumbai unit and Botany department of the college and said that Botany department has glorious history for its mycology research lab since 1976, which is recognized by UGC with research for mycological, and satisfactorily said that departmental faculties are following the path created by Honorable Principal Dr. V.R. Ghurde and Professor P.W. Devtare, who have devoted lot for the mycology. I would like to share here today's chief guest, Professor J. Tidke, former Pro Vice Chancellor, also completed his MPhil research from this botany department of this college in mycology. This workshop has included 4 technical sessions in which the renowned mycologists from our country have presented their talk on various topics of mycology. The unique feature of this workshop was conduction of assignment on one day and quiz competition was the second day to declare the winner and future mycologist. The first technical session was organized and it was chaired by Honorable Manoharachari and the speaker was Dr. T.S. Surya Narayanan, Director, Vivekananda Institute of Tropical Mycology, Chennai, who delivered a talk on the topic fungal endophyte and resilience of the plants to climate change. Dr. Surya Narayanan focused on the endophytic fungi and their relation to environmental changes with reference to application to the plant growth. In the second session, Dr. Sanjay Singh, scientist and coordinator, National Facility, NFCCI Agarkar Research Institute, Pune, delivered the talk on the topic taxonomy of hypomycetes fungi. Dr. S.K. Singh concentrated on diversity, morphotaxonomy, culture techniques, and technique of preservation of fungal cultures. He focused on the information of the NFCCI activities for mycology. Day 1 was concluded with assignment for the participants. We received huge number of participation from the student. On second day, Monday 13 November, Professor N.S. Atri, former President, MSI, Department of Botany, Punjabi University, Patiala, enlightened the participants on the topic morphological and anatomical parameters for characterization of lamellate mushroom. Professor Atri specially focused on the diversity and morphotaxonomy of the lamellate mushroom, which is burning topic of the mycology. He said that mushrooms are incredible creation of the God. The session was chaired by the Dr. T.N. Lakhanpal, Himachal Pradesh University, Simla. This was followed by the talk given by my friend Dr. Avneet Pal Singh, Managing Editor, Kavaka, MSI, Department of Botany, Punjabi University, Patiala, on the topic Systematics and Diversity of Polypores with particular reference to Northwest India. His major contribution in the poroid mushrooms he have presented. The session was chaired by Dr. Yashpal Sharma, Jammu University, Jammu. The assignment and quiz was conducted in which large number of participants wholeheartedly took part. The winner of the competition will be declared soon. The response of the student to this workshop indicated that the youth are having many innovative and creative ideas, and through this workshop, we have provided them the platform. In the form of workshop, we often provide them the unique chance to exhibit their ideas. The student's interest in fungal research is excellent, and we hope that upcoming future scientists may shine from this research fair of mycology. On behalf of Sri Shivaji Science College, I am thankful to the Mycological Society of India, Mumbai unit, for the collaboration of the event. We have tried our level best for the grand success of the event and hope we have succeeded in it. With this, I conclude National Workshop 2020, a remarkable, knowledgeable, and memorable event. We promise you all that in the coming years, we will again enter the world of mycology with some more innovative ideas to motivate and nourish your mind. This may be the end of this workshop, but actually, this is new beginning to apply the knowledge that we have gained through this workshop. Thank you. Thank you very much. Thank you.
Thank you, Dr. Dilip. It was nice to hear the entire journey of the workshop. Actually, this workshop was planned in the Association of Mycological Society of India, Mumbai unit. As rightly said by Dr. Dilip, this was the pressure on the organizers that how this entire organization will go. As the response was very huge, we have tried to satisfy all the expectations from the workshop. But as far as the collaborator of the event and the national organization working in the field of mycology, that is Mycological Society of India, and that too, the more prominent unit, Mumbai unit, was collaborating with this event. I think they are having huge expectations from whosoever is collaborating with them. We would like to know what exactly was the status of their expectations through this workshop. I'd like to request the secretary of the Mumbai unit, Dr. Sasirekha, to say a few words on this occasion. Dr. Sasirekha, please. Ma'am.
Yeah, thank you. I've respected members on the dais and the members online. It was a great experience through the 2 days of the online workshop. And I'm sure everybody agrees with me when I say we have definitely achieved what we expected in your college, that is Sri Shivaji Science College. And I must appreciate the efforts of each one of the organizing committee who has put in so much of efforts to make it a successful event. And definitely this online experience would have been much better when if we had in a different situation, like if we were physically been able to visit your university, it would have been even more enjoyable. But this was equally enjoyable for each one of us. And definitely I should place on record the appreciation for Dr. Dilip Hande and his team, who has taken so much of efforts right from the beginning of drafting the brochure till the execution of the entire workshop. It has been done so beautifully. And the definitely it all the credit also goes to the speakers who have been so very nicely and lucidly given us the details of each part of the topic so nicely that the students will definitely would have, if I have to say, got infected with the idea of doing mycology research. And I'm sure each one will enjoy doing this research without any fear of having any kind of adverse reactions. Like, normally fungi is believed, understood to have adverse reactions, but definitely the speakers have made it so comfortable that the researchers would definitely do it very well. And MSI Mumbai unit definitely would want to do collaborations with a number of colleges with more innovative ideas, and definitely want to spread the mycelium through the entire state, maybe later on the India, if I have to say. And we are greatly supported by MSI national body for this, because I definitely would like to acknowledge the appreciation and encouragement that we have received from the senior mycologists who have always been the backbone, I should say, or behind us, supporting us for these kind of activities, and that it has encouraged us to go forward with these activities. Definitely, this was just 2 days and 4 lectures. Maybe next time we would be having proper video sessions of the workshop, maybe. And this also can be a start of this thing, like making online videos for the students, where you can actually do virtual practical sessions which can be taught to the students. Like, they can this is specific for pandemic situation. Otherwise, obviously, everyone is doing a very good job in their own institutes. But if we have to continue like this, we can have these kind of things included. Otherwise, it was a very enjoyable experience throughout the 2 days. And I didn't feel that I was in Mumbai. I felt I was in Amravati. This organization was beautiful, and all the presenters have also done a very good job. And yeah, I think that that is the most, and I could say, and best wishes to for the collaborations henceforth continuously with mycology research. Thank you so much.
Thank you, Sasirika ma'am. It was wonderful remarks for us. It was the pleasurable moment for us. Actually, we have great potential to set the another milestones in future. We hope to have the same kind of collaboration from the MSI.
Yeah.
Specifically in the Mumbai unit, we have good relationships with all the people from the Mumbai as well as throughout the India. Whosoever is working with the MSI, they all are having the good relationships with the persons from the Amravati. So definitely we will set different events, and definitely this same response will be there for every activity we plan. Now, after hearing the remarks from MSI Secretary Mumbai unit, let me invite the participants who have participated in this workshop. I'd like to invite one of the participants, Mr. Prasad Pawar. Prasad Pawar from the Mumbai. He is from Zytex Biotech Private Limited, Mumbai. I invite him to give us feedback to improvise further in the next actions. Mr. Prasad, please unmute
Hello.
your mic.
Yeah. Am I audible? Hello. Is my voice audible to everyone?
Yes, you are audible.
Yeah. Yeah. Good afternoon, everyone. Myself, Mr. Prasad Pawar. I'm working as an assistant manager R&D in Zytex Biotech Private Limited, Mumbai. I would like to say thanks to all boarding committee members and organizers of Sri Shivaji Science College, Amravati, Department of Botany and Mycological Society of India, Mumbai Division, for giving me an opportunity to participate in such a beautiful workshop and also to interact with everyone regarding the feedback of this workshop. I really would like to say that entire workshop is very well organized. It is nicely conducted and it is very informative. I strongly feel that all the content of this workshop is very useful to all the students, um, faculty members, research scholars, and also to industrial professionals in the field of mycology. Online platform of this workshop is very easy to access anybody, and particularly this workshop is available on YouTube channel also, so that anybody can participate this workshop from his own place. So this is very useful thing for anyone. uh, one can easily find out the in-depth knowledge of speakers about the topics covered in all the sessions. It is also reflected in discussion session. So it is very useful for the budding scientists to understand the physiology, the morphology of the fungal spores and their attachment on hypha and another terminologies related to the mycology field. Mycology is a very interesting field, and I hope that this interest is further increased for all the participants who has attended this workshop. Personally, I mostly like the systematics and the taxonomical session of this workshop. After attending this session, my knowledge towards the identification and the classification of fungus has definitely improved. Last but not the least, about the assignments and the quiz competition. I really want to give hats off to the organizers who have prepared these assignments. The questions only tells us about the quality of this entire workshop. Overall, the entire workshop is very nice. It is very well organized, and in future also, I would like to participate in such workshop. I wish that the Mycological Society of India, Mumbai Division, as well as Department of Botany, Sri Shivaji Science College, that they further moving ahead, they can again organize such beautiful workshop. Once again, finally, I would like to say thanks to all the boarding committee members and the Mycological Society of India particularly to the Dr. Deshmukh, who has given me the chance to speak in front all of you. Thanks everyone.
Thank you, Mr. Prasad. It was the good remarks from you. We would be much more appreciate if the remarks are for improvement and suggestions to improvise the further events hosted by Sri Shivaji Science College, Amravati. We would like to hear a few comments from the another participant. We have with us Ms. Komal Verma from Department of Botany, University of Jammu. Ms. Komal Verma, please. Please unmute your mic. Komal Verma, please.
Hello. Hello. Good afternoon, sir. Sorry, this is Komal Verma. I am not Mr. This is
Yeah,
Mr.
so
Komal.
I must say sorry for that.
Okay, so it's okay. It's okay. Actually, I am very good afternoon to all of you, sir. I am very thankful to Shivaji College and MSI Unit Mumbai for giving me opportunity to speak something about this wonderful workshop. Yeah, all the sessions in this workshop are very well planned and they are very highly informative. The yesterday sessions by Professor Dr. T.S. Surya Narayan on fungal endophytes and the today sessions by and the two-day sessions by Dr. Avneet Pal and Professor N.S. Satri are very informative. I have learned a lot from this workshop, and definitely, I personally thankful to Professor N.S. Satri for giving me for giving such a wonderful presentation, and we have learned a lot from you, sir. And we have also we have learned how to identify what are the main characters we have to take in our mind to identify the mushrooms. One more important thing of this workshop is that all the assignments and the quiz that has conducted in this workshop are very. This is very positive point of this workshop. Actually, we actually what students have learned from the workshop. This will be uh, this will be displayed in this these quiz and those assignments. I highly congratulate Shri Shivaji Science College, Amravati and MSI unit, and also the organizing team to conducting such a wonderful workshop. And we request to you, sir, to conduct such workshops in the future so that all the candidates will be benefited. And thank you, sir. Thank you so much.
Mike. Thank you, Komal, for your remarks. It was really surprising for all the students who have participated. Many of the comments are there in the YouTube chat box also regarding the quality of the assignments and the questions placed in the quiz for assessing the workshop. I'd like to brief here that when we were planning the workshop, as it was labeled as the workshop, we were thinking that there should be some sort of activities students have to go through while going through this entire workshop. And accordingly, we have formulated the panel of experts from the state of Maharashtra, from different universities, and on the suggestions of all the experts from the field of mycology, we have planned the entire way to evaluate the total contribution of the participants. And accordingly, the quiz and the assignments were set. It was expected that the student, every participant from the workshop should work very hard during these 2 days, and he should do some activities. And because of that particular intention, we have placed some questions wherein students, all the participants have to go out of their homes and find out some sort of the specimens to upload the personal experiences in the activities which were planned in the assignment. Secondly, as far as the quiz is concerned, that quiz was also having the inclusion of the questions from all the 4 sessions. And it was not very easy quiz, like multiple choice questions and answers will be available from the Google. This quiz was completely based on the material given by resource persons in different sessions. And further, this was very necessarily requiring the work from the particular participants. And instead of going to the Google, you all must have visited the different books, Google, and all the resources wherein you will get the information. And mainly, I expect that entire information which you have placed, that must be from your experiences. And definitely, this was the main motto behind the organization of this kind of activities. And through these activities, it was the huge response from the participants. And through these activities, we have taken this activity as the competition, and that competition was for the 3 categories of the students and the participants. Those were UG students from the India, then postgraduate students from all the subjects throughout India, and the researchers conducting research for their doctoral purpose or for any kind of project or any research work. Amongst these three categories, we have compiled the data, score sheets from the assignment and the quiz, and summarizing that entire data, we have come up with the 3 prizes in each category. It is very tough competition, and the first and second and third prizes were with the margin of 1 or 2 marks. So it is very difficult for us to assess the entire assignments because that assignment was completely based on the activities. You all have to go through the activity and find out the answers for the question. And that was also having the multiple ticks in the same question. So it was the exhaustive work for all of you. I must thank all the participants and congratulate all the participants for successful completion of this assignment and the quiz. My dear friends, through the competition, we have shortlisted the participants on the basis of their scores, and merit certificates will be issued to all the winners. In every category, we have 3 winners, first, second, and third winners. May I now request Honorable President of MSI Mumbai Unit, Dr. Deshmukh sir, to please declare the winners before the entire August gathering. Dr. Deshmukh sir, please. Deshmukh sir, please unmute your mic. Deshmukh sir, please unmute your mic.
My mic is already unmuted. Dilip, you only tell the names. Dilip? Hello? Declare the names.
अभी मेरा ये जा रहा है, मेरा लॉग आउट हुआ अभी.
हेलो हेलो अनम्यूट के लिए लाए. हेलो 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 कैन यू हियर मी? हेलो.
यस सर, आ रही है, आवाज आ रही है.
बराबर है. दिलीप आप ही बता दीजिए नाम सबके. हेलो हेलो
लगता है दिलीप का म्यूट है.
एक मिनट
माइक
मैं बताता हूँ. I misplaced the file actually.
उनका कनेक्शन में कुछ प्रॉब्लम है शायद. दे आर गोइंग स्टिल.
हाँ या
वीडियो उनका स्टिल हो रहा है.
जस्ट 1 मिनट. हेलो.
आपके पास नाम दिया है क्या?
हाँ नाम है मेरे पास.
तो आप बोल दो ना.
एक्चुअली ठीक है. हाँ जस्ट 1 मिनट. We have names. We'll start from from research we'll start.
Okay sir.
Yeah. So Mr. Tokram Sarda Devi, Department of Botany, University of Delhi, first. Score is 66 out of 80. Then Megha Vekiram Gaura from University of Mysore, 54 out of 66. Dar Nadia Asraf, 50 out of 66 from Department of Botany, Kashmir University. Then Pushpa H from MS Rajaram College of Arts, Science and Commerce, 48 out of 66. This is the for research. For PG, we have H. Askaran from Pondicherry, Shankar Vaibhav Moreshwar from Mathura Das Mohita Science College, Nagpur. Pundir from University of Rajasthan, Jaipur. For undergraduate, Mrs. Preetha S. from Banrai Amman Institute of Technology. Second one is also L. Sapna from same institute. Then Akansha from Xavier College, Kolkata. And Kadu Arti Gajanan from RLT Science College, Akola.
ठीक है मेरा माइक अनम्यूट करना है हेलो सगळं बरोबर चाललं ना तर शेवटी So thank you very much for declaration of the results. It was the difficulty in the technical side because of which we could not display the certificates. Let me once again show the prizes to all the students because this may be having the lack of voice for students or the participants which are there on the Zoom platform. So let me share the certificates which are there in all the categories. This is the first UG category, and in the UG category we were having the multiple entries, and from that multiple entries first prize was won by Miss Preetha. Second, this first prize was shared by two students, and another student was Subri Sevilan. Second prize was for Shaw Akaricha, and third prize was to Miss Kadu Arti Gajanan. These are the three prizes from the UG category. Let me move on to the second, that is the PG category. And in the PG category, the first prize was won by A. Charuta Bhaskaran. Second prize in the PG category was won by Sakharkar Vaibhav Moreshwar. Third prize was to Pundir. These are the three prizes in the PG category. Let me announce the prizes from the research category. And in the research category, first prize was won by Thokcham Sarda Devi. Second prize goes to Megha Varshini Gowda. And the third prize was shared by two people. First one, Dar Nadia Ashraf, and second, Pushpa H. These were the prize winners, and I must congratulate all the winners for winning the prizes in different categories. So I'm also thankful to Dr. Deshmukh sir for declaring the prizes. Now moving forward in the valedictory function, I'd like to announce here that we have gone through different feedbacks from the students. Similarly, we are expected to have the feedback from all the students in different parts of India, because we would like to learn something out of this entire experience. And for that purpose, we have framed the feedback form, and that feedback form is available in the WhatsApp, Telegram, and our college website. So all the students, all the participants have must be submitting the Google form, and that Google form will be available on all these platforms. If you are not submitting the feedback form, we could not issue certificates to you. Please take care and take a note that whatever email ID you will use while submitting the feedback form, the same email ID you need to use for downloading the certificate. To download the certificate, link is available on the college website, from where you have downloaded the different links for YouTube streaming or feedback or quiz or assignment. So, dear friends, your feedback is valuable for all of us. That's why I request all the participants on the YouTube and those who are registered for this workshop to submit the feedback in time, because by tomorrow morning 12 noon we will issue the certificates from the same certificate portal. This certificate will be issued to only those students who can submit their feedback before 12 midnight today. This night is the last time for you to submit the feedback. This was the instruction, very important instruction for all of you to get the certificates from the organizers. Now, we are very fortunate to have the dynamic personality amongst us as the chief guest for this valedictory function. We have with us Professor Dr. Jaikiran Tidke, who is head professor and head department of botany, Sant Gadge Baba Amravati University, Amravati. And we especially the faculties from the department of botany, Sri Shivaji Science College, Amravati, are blessed with his guidance, all-time guidance, because we all are the student of Dr. J. Tidke sir. He has long experience of 26 years in teaching and 11 years in the administration, because he has honored the chairs of the Pro Vice Chancellor and Vice Chancellor as well. So under his able guidance and leadership as the Pro Vice Chancellor, entire track of the research in the field of sciences, he has revolutionized the entire way of the research when he was the Pro Vice Chancellor of the Amravati University, and being the chairman of the RRCs in variety of the areas of the science, he has revolutionized the complete way of research in Sant Gadge Baba Amravati University, Amravati. While mentioning his area of research, I must say that he is having the huge contribution in the field of palynology. He is working on the pollination biology, plant reproductive biology, aerobiology, pollen biotechnology, and the more modern area of the bioinformatics. His research experience is very huge. Since last 23 years, he is contributing, consistently contributing in the area of his interest. Many of the students achieved PhD under his guidance. I must mention the number here. He has guided 15 students for the doctoral research, and all the students awarded with the degree. As far as the publications are concerned, Dr. Tidke has one patent to his credit, 54 research papers published in the national and international journals, 104 conferences he has attended, and he has organized 5 conferences as the convener. He edited one book, and in the different universities, he has evaluated different thesis, PhD thesis in the area of palynology. He has visited many countries for academic deliberations. I must mention he has visited the Hafia University, Israel, Vienna University, Austria, Institute for Botanic, Vienna University, Vienna, Vienna University, Botanical Garden, Oxford University, Oxford, UK, London University, London, Kew Botanical Garden, London. This experience he has collected from the globe, he disseminated it in the entire fraternity of the botany. We are the recipient of his advice. Thank you very much, sir, for being with us in this evening for the concluding ceremony of the national workshop. May I now request Honorable Chairman of this session, Dr. H.S. Lunge, sir, to acknowledge the contribution of the chief guest of the session, Dr. J. Tidke, sir, in the botany department, in our personal career, and in the field of botany as whole. I request Honorable Dr. Tidke, sir, to kindly accept the honor from our IQAC coordinator, Dr. H.S. Lunge, sir. Thank you very much, sir. May I now invite Dr. J. Tidke, sir, to bless us with his guidance. Please, sir.
I can say good evening to everyone now. Dear friends, at the outset, let me extend my sincere thanks to the organizer for calling me here as a guest for validity function of workshop on fungal systematics and technological advances. On the occasion of valedictory function, in the chair, my friend Dr. S.S. Lunge, head of the department Dr. Dorkar, organizing secretary Dr. Hedau, another organizing secretary Dr. Dilip Hande, and all respected colleagues present, the president of MSI, the secretary, and the delegates, I can say the e-delegates who are present, who are attending this function and who attended the whole event since yesterday. So to organize the conference is a very difficult task. And this is one of the premier institute in Vidarbha region, that is Department of Botany, Sri Shivaji Science College, Amravati. It's a unique one, unique in the sense during the early time, say 70s or 75, this particular department has the recognized mycology lab under the leadership of then Principal Dr. V.R. Gurude. And I was fortunate to work with Dr. Gurude as he was my guide here for MPhil dissertation. And I did work on the soil fungi. So behind all the efforts by late Dr. Panjabrao Bhausab Deshmukh and then the principals of this college, the Department of Botany is doing well in the field of the mycology. The team of young mycologists is here in this department and they are doing quality work and contributing to the mycology subject. When I was listening to inaugural talk delivered by Dr. M.K. Rai, he said that maybe in any field of botany, say algae, fungi, bryophyte, pteridophyte, gymnospore, angiosperm, taxonomy is most ignored subject. Nobody liked to work on taxonomy. And when you need the organism, you should know the name and systematic position of that particular organism to work out the different aspects of the organism. So he told that taxonomy, the people take it as a tax on me. Yesterday I was listening, but it is not like that. The basic mycology deals with the taxonomic work of the fungal organism. And the fungal organism or the fungi, they are such a organisms at every corner of the life they are touching. Maybe life of the plant, life of the microorganism, life of the human being. They are involved in so many industries. And when there was an epidemic, say 70, 80 years ago, we have the substance which was extracted from the fungi itself, that is the antibiotics. And all these antibiotics, since that time, they are working to control the human diseases. I hope that during the deliberations of this conference, the workshop, one might have been came across to find out the remedy for COVID-19. Hope in future the fungi will come out with some element or with some drug or some medicine which will be extracted from fungal organism itself, like the antibiotics which were extracted, which were obtained from the aspergillus. Maybe a possibility. The new researchers, because it's a huge gathering of e-delegates, more than 1,000 delegates are there, or somebody may be there who can try the fungal organisms to control the COVID-19. Always there is possibility. We can't deny the possibilities. So this will be the tribute to the people who have established this esteemed institution. And let me put on the record my heartfelt appreciations towards the organizers, Dr. Hande, Dr. Hedau, Dr. Tushar, Magirwar Madam, and the head of department, Dorkar sir and Dinesh Khedkar. They are restlessly working for the success of this conference, because to organize the conference is not a easy task, and everybody might have been gone through the work done by this team, and everything is done very systematically. Everything is very systematic, and the schedule of the conference was also observed systematically. Everything goes smoothly. And in the age of the technology, what is important, if you see the title of the workshop, it is also very interesting. What is the title? Fungal systematics and technological advances. Though systematics, that is taxonomy, deals with the basic aspects, we have to join these basic aspects with the technologies, and that to the advanced technology. That means one has to use the advanced technologies to solve the problems related to fungal organisms. So it is nothing but the cohesion of two things, or the blend of two thoughts as per the requirement of the new areas of the research. We should go for the recent techniques or advanced techniques to solve all the problems related to fungi, or in general related to plant and animal, including the human being. I'm happy to be here because being a secretary of Indian Aerobiological Society, and I am also the life member of Mycological Society of India since long time. Most of my friends, aerobiologists, they are member of the Mycological Society of India. And the team from the Amravati region, that is from Sant Garge Baba Amravati University, we all the people in collaboration. It was during my tenure of Pro Vice Chancellor, we had the collaboration 2012, collaboration with Department of Botany and Mycological Society and Department of Botany, Sri Shivaji Science College, Amravati. That time, 435 delegates were there. All the stalwart mycologists, they visited our university and this college. This was the giant venture, and it was grand successful. We were doubtful about the gathering that time also, but the response was so huge, and this time it crossed the four digits, 1100. So what we are doing here in our department, that will go to the people throughout the India, and that will speak about the quality of the teaching and research in our college, in our institution, or in our department. Whatever output of this conference will be there, that will be reflected in the form of the work to be undertaken by the new researchers. So quality will always speak. I think those people who have attended this particular workshop, they will appreciate your efforts. And if you see the souvenir abstract book, lot of new concepts are there, and lot of things are there which can be inculcated in the new generation. And about the standard, or say the quality of the work, if you have the quality, the people will take care of you. If you don't have the quality, nobody is going to take care of you. So once you adopt the quality mission in in your work, research, or by all means, if you have the qualities, the society has to appreciate it and it has to be reflected through your work. So this team did very nice work, and in future also, I hope there will be more collaborations for undertaking the work related to mycology. My team is always with you. No doubt we are on the verge of contributing lot of things. Lot of things has to be come, and you have to put all these things in the form of the standard publications, and that will be appreciated by the people. What taxonomists look for, that is the basic thing in this workshop. The taxonomics always look for the character. If the character or the character from the different sites they are collected, then and only then you can identify the species. The identification of the species, naming of the species is not merely the meaning of the taxonomy. It includes the techniques, it includes the advanced techniques, rather to say. And by using advanced techniques, one has to, or the mycologist has to find out the character. And by using this character, we can have the similarities and differences in the morphological structure, genetical structure, or say in habitat and habit of the organism pertaining to fungal group. And once you have the character, you have to compare it. And when we compare, we look for the character. It is obvious that when we want the different characters, we are looking for similarities. When we have to study the similarities, we have to look for differences. That is the strategy. When you go to market to buy some sari, which will be identical to the earlier one, what for we are looking? We are looking for differences. We want similar saris, but we look for the differences. That is the thing. This particular principle operates here also. If you have different characters, only and only then you can have new group, new organism, new species, new things. So this is the scenario in case of the taxonomy. Ultimately, one has to find out the characters. So to find out the characters, you have the new technologies, and not only the characters, you have to go for some elements, some products, some byproducts, or a lot of products which are obtained from the fungal organism, secondary metabolites, and lot of things are there. So by using all these techniques, we can have different areas or advanced areas of the research in our institution. I should again appreciate the efforts made by the Honorable Principal of this college, the head of the department, and the co-workers team of mycologists from this region. Again, I put my sincere appreciations and conclude my speech. Lastly, extend my sincere thanks to Sri Shivaji Education Society, principal of this college, and the organizers. Thank you very much.
Thanks a lot, sir, for kind words of appreciation. Your words of appreciation will definitely boost our morale to host such kind of magnificent events hereafter. So this was the workshop which was planned to have more focus on the fungal systematics that was based on the taxonomy. In our discussion also, we were having the same line of thought that it should be coming to the molecular level. And for that purpose only, we have added the technological advances. And these technological advances are the thing which we expect from the students, from the teachers. As far as these two conventional and modern extreme ends are concerned, this is just the effort to provide the simple, pleasurable experience, not only the measurable learning thing. So to make the measurable environment pleasurable, we have planned different activities. And we are very fortunate. We are very lucky enough to have third collaboration coming into play in this organization. We are having one more, say, invitation from the very renowned institution of the India, that is Agarkar Research Institute. I'd like to invite Dr. Deshmukh sir for briefing regarding the this third angle in the workshop, and say few words, because in the earlier interventions, because of the technical flaw, we could not hear Dr. Sunil Kumar Deshmukh very lucidly. So I request Dr. Sunil Kumar to throw light on this interaction with the Agarkar Research Institute, MSI, and the Sri Shivaji Science College. Deshmukh sir.
Actually, now we are coming to the end of the 2 days workshop. I hope you got benefited with the presentations by the eminent speakers and might have gained the knowledge for future research. Here I would like to tell you, when I was working with the mushrooms from Medghat, we have got a large number of diversity from there and large number of bioactive metabolites. Hope you people will be taking a chance to learn more on that, because my stay and that area was limited, and because of monsoon condition, you cannot continue working over there. And continuing to that, the winner of the quiz contest will get a chance to interact with the scientist of NFCI Pune at Agarkar Institute and get hand-on training of fungal identification and bioprospectings. This fungal identification will be having both taxonomical, means classical, along with the biochemical and molecular. So if you have hand-to-hand both the identification, then you can be sure that this is that particular fungus which you are calling as a new. In past, we have said this is a new species from India, and when other persons are working on monograph, most of the species were merged. So with the molecular tool, this is very sure that what we are reporting is correct. The formal invitation will be sent by Director, Agarkar Institute, Pune, in due course, and organizer will inform the recipient in due course of time. And at the end, I would like to thank thanks the Shivaji Science College for organizing this. Coming to the MSI Mumbai unit, we will be additionally organizing webinars on different topics. We have another series of the presenters, basically the postdocs and the young teachers who are working on the bench, and we would like that students should have the idea what they are working. They should have the networking with the similar group. Then we will be having one workshop by the photographer by National Geographic, Dr. Ru, who will be telling how he is engaged in the taking photographs in the nature, particularly mushrooms and the family Gileriaceae. Another with that, we will be having the lecture series on the writing skills, because most of the papers what we see, they are got rejected because of the low quality of the writing skill by most of the students. And we are publishing the paper in local journals with low impact factor or no impact factor. So we will be having some program with Maharashtra Academy of Sciences, particularly for rural people. As a member of or the fellow of the Maharashtra Association of Society, Maharashtra Academic Science Society, I had discussion with them several times. So if rural area people are interested, we can have the collaboration with this aspect. And at last, I am thankful to all of you for organizing wonderful workshop.
Thank you, sir. Thank you very much for your kind remarks. How wonderful this event is. Whosoever is participating and winning the prizes is given away with one more opportunity to work in the national institute like Agarkar Research Institute, which is very reputed institution for the studies in mycology. So dear friends, dear student friends, here this kind of workshops where all the quality parameters has taken care of the difficulty level is very high. In such kind of workshops or such kind of events, you need to come forward and participate, because to get the huge round of applause in the stadium, you need to hit the difficult ball instead of the normal and routine ball. So such kind of workshops will definitely give you enriching experiences, and similar experience is given by Sri Shivaji Science College, and that too when we are having the collaborator like MSI. Definitely the stadium will be roaming with the students who are having the laurels with their credits. So thank you very much to all the participants. Now I invite chairman of this session, Dr. H.S. Lunge sir, who is IQAC coordinator for the college, to deliver his presidential remark.
Thank you, sir. Today National Online Workshop on the Fungal Systematics and Technological Advances. This is the valedictory function of this two-day National Workshop. For this valedictory function, we have the chief guest, our friend, Pro Vice Chancellor, former Pro Vice Chancellor of the Santa Gadge Baba Amravati University and head of the Department of Botany, Santa Gadge Baba Amravati University, Amravati. The head of the department, our friend, Dorkar sir, the organizing secretary, head of sir, Hande sir, Dr. Dinesh Khedkar, the team of the botany department, Wankhede sir, Magirwar madam, Darsimbe sir, and the Punkar madam. It's very, very appreciable thing. Today, it's very fortunate for us that in this pandemic COVID-19 period, this workshop has organized by the Department of Botany. And it is, I feel it's my fortunate that the Botany Department constantly getting the inspiration and the support and the cooperation from the Professor Tidke sir for all types of the academic work. The team of the Botany Department working very hard. This workshop was organized very systematically. I feel that it is not only the online workshop, but it is the combination of the offline and the online workshop. And the another thing I must appreciate on behalf of the principal sir, on behalf of the all stakeholders of the botany and the botanical persons of the Amravati region, and from on behalf of the IQAC, that the Mycological Society of India, the Mumbai region, Professor Deshmukh sir, Secretary Rekha ma'am, and their executive members all have given the not only the collaboration, joint collaboration, but also continuously support. The support not only on the paper, but academically support. Continuously they are here from yesterday, from the inauguration function and also up to the valedictory function. This online workshop organized on this new topic and with the technological advances, no doubt, it is very, very useful for the new generations to come to work for the recent and the new research work. It is very, very happy to listen that within this workshop, the quiz, the three types of the quizzes for the UG people, for the PG people, and for the research students. And the names I have heard here, the winners that was from all the corners of the India, and the participants, 1100 participants, more than the 1100 participants from the national and the abroad, India and the abroad. So it is the more success of this workshop organized by the Botany Department with the help and the collaborations and the guidance from our friend, the Professor Tilke sir, and on all the botanical persons. I must appreciate the teamwork of the botany department under the leadership of Dorkar sir, all the teachers, the non-teaching staff, on behalf of the IQAC, on behalf of the principal sir, and all stakeholders of the Sri Shivaji Science College. I congratulate the Department of Botany, and I must express my sincere thanks towards the Professor Deshmukh sir, the president of the Mycological Society of India Bombay unit, and his team, who has constantly and continuously helped in an academic manner, and also the resource persons from all over the India and abroad also. And also I appreciate the listening patience of the participants from yesterday, whosoever the participants listening this workshop, all the technical sessions, no doubt it was a very quality level workshop, and all the topics, all the resource persons has contributed very well in a micro level about the particular subject. I do not know about the this botany, but just I know that only the in all the field, no doubt today there is a work of research work in the field of the interdisciplinary way. So computational biology is also the new recent topics which will be help for the new researchers. I personally and on behalf of the principal sir, I express my thanks towards the Professor Tilke sir that he has spared his time and express here his thoughts about this theme of the workshop. I also congratulate all the team, and I also congratulate and thanks to the Professor Deshmukh sir and his team. Okay. Also thankful to the organizer, the calling me here for this valedictory function and giving me the chance. Thank you. Thank you.
Thank you very much, Dr. Lunge sir, for appreciating us for a variety of the efforts we are taking in a team. My dear friends, as I have already said to you, we are in the transforming state of education. In earlier days, our complete learning was memory based. As far as today's scenario is concerned, we all are knowing that while purchasing any kind of mobile, instead of looking after the memory, we used to look after the processor. And as far as the students are concerned, researchers are concerned, you all should focus on the processor, which should be hardcore processor, which should be the multi-level processor, and that processor will be in your mind, and that mind should not drag towards the memory. That mind should drag towards the actual experience. And in this workshop, one benefit is given by Dr. S.K. Singh sir, and that benefit is for every winner in the category of UG, PG, and research student. Top winner will have the actual experience at the NFCCI Pune, and this is the laboratory of Dr. S.K. Singh, and this is the lending hand from Dr. Sanjay Singh. He is inviting the top performer from this workshop to learn, to get the actual experience of research at this National Institute. My dear friends, this is the wonderful opportunity for you people. And those who are not having that kind of position, but still you will have the huge learning experience, and that entire experience is placed on YouTube platform of our department. You can visit these sessions at any time. All the sessions will be there on the YouTube platform. All the links for the feedback, for the recorded session, for the live streaming are there on the website of the college. So please visit our college website and do submit feedback form, because that feedback form will make it easy for you to download your certificate very easily. And I once again repeat here that the certificates will be issued only after tomorrow 12 noon. before tonight, 12 midnight, you all have to submit your feedback at the given link. That link will be active only after finishing this valedictory function. So these are the things which you should take care of to get the certificates. All the participants, all the teachers and participants who will submit the feedback form will get the certificate. Otherwise, if you will not be able to submit the feedback form, it will be difficult for us to issue the certificate because of the database based mechanism of the certificate distribution. So coming to the end of this session, I am inviting Organizing Secretary Dr. Ganesh Hedau to propose vote of thanks for the entire workshop.
Thank you, Dinesh. Good evening, everybody. Honorable Chairman of Valedictory Function, the Chief Guest, Office Bearers of MSI, Senior Mycologists, Honorable Chairmen of the various sessions, guest speakers, professors, teachers, industry persons, research scholars, and dear students of the different part of the world. In the capacity of an organizing secretary of this 2 days national online workshop on fungal systematics and technological advances organized by the Department of Botany, Sri Shivaji Science College, Amravati, in association with Mycological Society of India, Mumbai unit, I feel proud and privileged to propose the vote of thanks. At the outset, we express our gratitude to Honorable Dr. Sunil Deshmukh, President MSI Mumbai unit, and the executive members for the alliance with our college to organize this unique event in the greater way. Dear participants and respected invitees, while searching for the most important contribution for this event, with whom I must begin to acknowledge to its Honorable H.S. Lunge, IQSC coordinator of Sri Shivaji Science College, Amravati, has remained present as a chairperson for today's valedictory function. We are very fortunate to have a very good administrator, passionate and the charming personality in the field of botany, Professor J. Tidke, head of the Department of Botany and ex-pro vice chancellor of the University. So, we're highly grateful to you for your guidance and presence. I'm highly grateful to Dr. Vijay Thakre, principal of the Sri Shivaji Science College, Amravati, for being chairman for the inaugural ceremony. The words of gratitude definitely will not be enough to acknowledge you, sir, as you have constantly supported us to organize this event. The event would have been incomplete without the presence of Honorable Professor M.K. Rai, Basic Science Research Faculty Fellow, Department of Biotechnology, Sant Gadge Baba Amravati University. We are indebted to you for inaugurating this workshop and your valuable address. Let me acknowledge all the chairmen and the speakers of various sessions for these 2 days. Firstly, I would like to express my sincere thanks to Honorable T.S. Suryanarayanan, Director of the Vivekananda Institute of Tropical Mycology, Chennai, Honorable S.K. Singh, Scientist and Coordinator, Agricultural Research Institute, Pune, Honorable Professor N.S. Satri, ex-professor, Department of Botany, Punjab University, Patiala, and Honorable Dr. Avneet Pal Singh, Department of Botany, Punjab University, Patiala, for gracing their crucial work and sharing with us their valuable opinions and findings. Let me record our deep sense of gratitude towards all the chairmen for sparing their valuable time. Professor C. Manorachari, Dr. Shilpa Virekar, Professor T.N. Lakhanpal, and Professor Yashpal Sharma, who have kindly consented to chair the session and expressing their remarks during this workshop. We must acknowledge the charitable publicity offered by paper and electronic media to draw attention of the common citizens of this region towards such a magnificent event organized by our department. We are grateful especially to the members of the local advisory committee and MSI members of Amravati region. I heartily congratulate to the winners and all the participants for their active participation in this event. I thank to our distinguished guests, experts, and the students who have been kind enough to join us online and valued scientific deliberations by sharing their research experience. Finally, I extend my thanks to our head of the department, Professor B.K. Dorkar, co-convener, and all the organizing committee members and non-teaching staff for their enormous cooperation in the organization of this workshop. I hope this workshop will guide us on many counts. Dear participants, we hope to have your enormous response in future also for such academic ventures. On behalf of the president of this valedictory function, I declare that this workshop is over. Thank you. Thank you very much.
Thank you very much to all the members in the Zoom meeting. And I must thank all the dignitaries on the dais. That we come to an end of the meeting. Thank you. Shall I end the meeting? Yes. Thank you. Thank you very much.
Thank you.